Welcome back, students. This is my fifth video in my YouTube channel. In my last four videos, I completed the first chapter of class 12, reproduction in organism. Now this video I am going to start with the second chapter, that is the sexual reproduction in flowering plants. Okay, so let's write the name of the chapter here, that is sexual reproduction in flowering plants. Okay, so please see this video till the end, okay, so that whatever we will discuss, the topic, the concept will be clear. I will try my level best to explain each and every details, whichever is important. And the most important thing is that this video, this topic that I am going to discuss, this is totally for the board students. Okay, this is not the entrance level discussion I am doing. Okay, that will be a separate video for the entrance level. This is only the board based explanation of the topics. Fine. So some basic informations first. Sexual reproduction in flowering plants. The flowering plants means we are talking about the kingdom angiosperms. Angiosperms. Okay, so this is the division of the plant kingdom under which all the flowering plants come. So in these angiosperms, flower is the reproductive organ. So first information, the reproductive organ in these angiosperms are the flowers. Okay. Now we will be getting inside of flower. Okay. Parts almost all of us have idea, but still just a revision. Number one, we are dividing the parts of the flower into two different types. One is essential parts, and number two, non-essential parts. Okay, the essential parts are stamens and carpels, which is also known as pistils, and non-essential parts are sepals and petals. I expect all of you to jot down whatever important you feel on the board. Okay, so sepals and petals. Sepals, the group of sepals are known as calyx. Group of petal is known as corolla. These are the biological terms which are important. Now stamens. Stamens are the male reproductive organ in the flower. So the group of stamens are known as androecium. The spelling is also important. And the carpels are the pistils, which are the female reproductive organ in a flower. The group of carpels known as gynoecium. Okay, so these are the four different parts of a flower. Now we will see our concern in this chapter is the stamens and carpels. We will discuss about this. Okay, the non-essential parts we have already studied in class 11. Stamens. What is the basic structure of a stamen? Okay, this is just an idea how the stamen look like. Okay, so this part, this is the filament, and this part is the anther. Okay, now little details. So stamen contain two important parts, filament and anther. Now little details of this anther. Okay, we have to know the detailed structure of this anther and the functioning of the various parts. This part is very important for us. Some informations. Number one, about the anther. About the anther we are going to discuss now. Okay, this anther, it is number one, bilobed in nature. Bilobed in nature. Okay, two lobes are there. Okay, this lobe, one side you can see, another one, this one I have drawn at the back. So two side by side, two lobes are there. Okay, so bilobed. Number two, dithecous. Dithecous. Dithecous means di means two. Two theca. Theca. Theca means chambers. Okay. It means that two chambers are there. Okay. It's like this. If you are dividing one lobe longitudinally, okay, it becomes two chambers. Okay. Chamber 1, chamber 2. Understood? Same on that lobe also. Okay. So two chamber or two theca we get. So ultimately 4 theca altogether in an anther. We see how many? 4 theca. 4 chambers are there externally. Okay. This is some structural feature of anther. Now, if I observe the transverse section of this anther, transverse section, like if I cut it like in this way, if I cut it like in this way, transverse section, then how the anther inside, how the anther would look like. Okay, that is what we will see now. I am drawing here. This would look like this. After transverse section, it means this is an anther, for example. And if I do this transverse section like this, then how it will look like the inside. The picture is like this. Okay. Now these are, see, two lobe I said. So one lobe, another lobe. Okay, and each lobe. Okay, I say two chambers means two theca, 1, 2, and this lobe 1, 2. So 4 theca I can see. So 2 lobe. So if I like this, if I divide it like this, okay, it will be easier for you to understand. So this side lobe, this one lobe, this whole one lobe, and this is the another side. Okay. So each lobe having 2 theca. So 1, 2, and this lobe having 2 theca. 1, 2. So then 4 theca here. 4 chambers. Now we have to know little details of this structure also, which is important. Okay. From this, I am just rounding this part. Okay. If I magnify this area, then how this will look like. Okay, starting from the outer to inside. Okay, starting from the outer to inside, how it will look like. I hope you are jotting down the important terms. Okay. Just I'm trying to give you an idea how these layers are formed in this anther from outside to inside. Okay. This is the number 3 layer, third layer I am drawing here. This is the innermost layer I am drawing here. Okay. So I hope it is clear the which part we are actually seeing, this area. Okay. Now see, let's level it. Yeah. This is the outermost layer. You can see this outermost layer. This is known as the epidermis. 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 Epi means outer part and dermis means layer. So epidermis is the outer layer. This is the second layer. Okay, so this one is known as the endothecium, second layer. Okay, there is little one more layer has to be here. Just 1 second. Little modification in this diagram because there are altogether 4 layers you see there. Okay. Fourth layer, which is the innermost layer. Okay. Fine. Now, so first the outermost layer is the epidermis, then we see the endothecium, the second layer, the third layer I am writing here is known as the middle layer, and this innermost, okay, the innermost, let's label here, is the tapetum. Okay, so these are the 4 layers. Number 1 is the outermost layer, epidermis. Second one is the endothecium. Third one is the middle layer, and the innermost layer is the tapetum. They are the 4 layers. Okay. Now let me tell the function of this. Okay, this epidermis, the endothecium, and the middle layer means the outer 3 layers. Okay, let's number it. Number 1, this is number 2 layer. This is number 3 layer. This is number 4 layer. The outermost 3 layers protect the cells inside. Okay. And the innermost layer, which is the tapetum cells, okay, this cell provides nutrients. Okay, provide nutrients. Nutrients to whom? Nutrients to, there are cells present inside this chamber. There are cells present inside these chambers. Okay, so these tapetum cells, they provide nourishment to these cells. There are names, particular names are there. Now let me tell you, this whole chamber that I have shown here, this chamber, the whole chamber is known as microsporangium. Okay, microsporangium. And the cells inside is known as sporogenous tissues. Sporogenous tissues, which are also known as or microspore mother cell. Okay, microspore mean actually it is talking about the pollen grains. So it is MMC. Okay, or it is also known as pollen mother cell. So PMC, the same thing. Okay, the sporogenous tissues or microspore mother cell MMC or pollen mother cell PMC, the same thing. Okay, so these cells will grow. So for that purpose it needs nutrients. So the nourishment is provided by the tapetal cells here, the innermost layer. One specific feature of this innermost layer, tapetum, is that these cells are, I am rubbing this part. Okay, these tapetal cells are multinucleate in nature. Okay, which is also known as syncytium in nature. That is, the cells are multinucleate in nature. Okay, so this is the structure. This is the microsporangium here. The chambers are known as microsporangium. This is the layer structure. This labeling is very, very important for your exam point of view. Okay, so this part we have learned here. Clear? Now the next part, this sporogenous tissues. About this structure is very, very important. This, whatever I have drawn the labeling, you have to learn this, you have to practice first, okay, with the labeling. Now these sporogenous tissues, okay, the sporogenous tissues will undergo actually meiosis cell division. The sporogenous tissues will undergo meiosis cell division. So each sporogenous tissue or each microspore mother cell or pollen mother cell PMC, okay, are going to give rise 4 new cells, total cells 1, 2, 3, 4. Okay, these are the pollen grains, which is known as microspore. Okay, so that's why the pollen grains, the microspore, that's why this cell is known as microspore mother cell, from which the microspore developed, or pollen mother cell because pollen grain developed. Okay, so from each sporogenous tissue, okay, 4 pollen grains are going to develop. This pollen grain, biologically, technically, it is nothing but male gametophyte. This is very, very important to remember. This is a male gametophyte. The pollen grains are not male gametes. The pollen grains are male gametophytes. Okay. Now, what is this? What are these gametophytes actually? Gametophytes are the structures that give rise to gametes. It means the pollen grains are the male gametophytes, and these are going to produce or develop male gametes. Okay. Now, question come on this topic. Question comes. How? I am rubbing this structure. Okay. Suppose, one example, if a microsporangium, if a microsporangium contains 200 sporogenous tissues or MMC or PMC, any term can be there in the question. So if a single microsporangium contains 200 sporogenous tissues or microspore mother cell, how many pollen grains? The question is how many pollen grains will be produced by this anther. Read this question carefully. A single microsporangium contains 200 sporogenous tissues or MMC. Then how many pollen grains will be produced by this anther? If you think little, okay, we'll get the answer. See, one single microsporangium contains 200 sporogenous tissues, right? So the question is how many pollen grains will be in an anther. So one anther contains how many pollen grains? 4. Fine. Sorry, microsporangium 4. So one microsporangium having 200 sporogenous tissues, right? So I have shown here the one sporogenous tissue undergo meiosis cell division produces 4 pollen grains. So 200 into 4, 800 pollen grains are going to produce by 200 sporogenous tissues. It means one microsporangium produces 800 pollen grains. Now there are how many microsporangium? 4 microsporangium are there. So it means that into 4, 3200 pollen grains will be produced by an anther. When it is talking about anther, it means it has 4 microsporangium. So one microsporangium produces 800 pollen grains. So 4 microsporangium produces 800, 800, 800, 800. So it's 3200 pollen grains. Okay, so this kind of questions may come 1 marks or 2 marks questions. Okay, now next our so this is done. Now we have seen here that pollen grain is produced, right? Here pollen grain is produced by meiosis cell division. Okay, now we will see the structure of pollen grain now. Okay, structure of a pollen grain. Structure of a pollen grain. See, the outermost layer of a pollen grain is known as exine. E-X-I-N-E, exine. Okay, this exine is made up of a substance known as sporopollenin. Okay, the exine, the outermost layer, is made up of a substance known as sporopollenin. And what is this sporopollenin actually? Okay, it is the hardest and most resistant organic substance ever found. It is so tough component, okay, which is present in the exine. It is the most resistant organic substance ever found. It means that extreme acidic condition, extreme alkaline condition, okay, cannot damage this exine because of this sporopollenin. Okay, even enzymes, no enzymes still that found that can damage this sporopollenin or in turn to the exine. So you can understand that how protective this sporopollenin or the exine, which is the outermost layer of a pollen grain. and this is because of the presence of this sporopollenin. Okay, we found the fossils of pollen grain in a so good condition. Okay, because of this sporopollenin. Okay, and wherever we see the fossils of pollen grain, that to some extent confirms the presence of petroleum or coal in that area. Now this is the outermost layer. Okay, this side I am having now. The innermost layer, the inner layer is the intine. Okay, which is made up of cellulose and pectin. These are the polysaccharides, cellulose and pectin. Now this is a thin layer. Okay, there are some areas, okay, around the pollen grain. Okay, suppose this is a pollen grain. Some areas the exile becomes very thin layer or absent. Some areas. So just like for example, this area it is absent. Just let's see, this area it is absent. Okay, or some area it becomes very thin. Some area again it is absent. Okay, absent, and like this it happens. Okay, some area. So these areas where it is absent or it is a very thin layer, these areas are known as germ pores. Okay, and suppose this is the inner layer which I have drawn already. This is the intine or the inner layer. Okay, during germination of the pollen grain, what happens is that this intine. Okay, suppose there is a pore, again one germ pore we can see. So during germination of the pollen grain, what happened is that this intine come out. This intine come out in the form of pollen tube. The intine come out in the form of pollen tube. Okay, we will see later on the pollen tube development. Okay, after pollination. So this concept has to be there in your mind. Okay, so the exile, the germ pore and the intine then come out in the form of pollen tube from the pollen grain during germination. Okay, fine. Now exile, intine we have done here. Now we will see the shading of the pollen grain. Okay, shading of pollen grains. See what happens is that, let me, before getting into the shading part, as a pollen grain, suppose it's a pollen grain contains initially one nucleus. Okay, initially one nucleus. Now this nucleus undergo unequal mitotic cell division. Okay, as a result of which what will happen? This pollen grain will be having two unequal nucleus because it is unequal mitotic cell division. So one nucleus divided into two, but these are unequal, one large and one small nucleus. Okay, and both are present in the cytoplasm. They are surrounded by cytoplasm. This small nucleus is known as generative nucleus, and the large nucleus is known as vegetative nucleus or tube nucleus. Okay, now what happens? These nuclei, both these nuclei, they surrounded by cytoplasm. Okay, cytoplasm dense, because what happens is that first of all karyokinesis, do you know the karyokinesis and cytokinesis before cell division? So that, I mean, these are the parts of cell division. So before this is karyokinesis happened, means nuclear division happened. Now after karyokinesis, the next step is cytokinesis, means cytoplasm division. So karyokinesis done, nuclear division done, though here it is unequal. Now cytokinesis will happen, cytoplasmic division, as a result of which what will happen? This nucleus will get surrounded by a separate cytoplasm here, and this is also getting a separate cytoplasm inside. As a result of which now we say that this is, now it becomes one cell, this becomes another cell. Okay, so now this is cell, this large cell is known as vegetative cell. Okay, or we say it is a tube cell. I am just showing this separation. The picture is not exactly like this. There is a difference. Just to make you understand, I am putting the image in this form. Okay, it is actually different little. So this is tube cell, and this is, I will say, generative cell. Okay. Now see, at this stage, this pollen grain contains two cells, vegetative cell and generative cell. If the pollen grain shed at this stage, it is known as two-celled stage of shedding. So the pollen grain sheds. If it shed at this stage, shed means it's releasing out from the anther. Anther dehiscence. Anther dehiscence means cracking. So cracking of the layer, the middle layer, the endothecium, epidermis we have studied. So the cracking of the layer, that's called dehiscence of anther, and as a result of which the pollen grain will be released. That is called shedding. So in 60% case, 60% of plants, we see the two-cell stage shedding of pollen grain. In 40% plant case, we see three-cell stage of cell division. The pollen grain shedding. Okay, three-cell stage means what? This generative cell. Okay, so let me draw here. This is the vegetative cell I am getting here. A generative cell. Generative cell here, it divides into two male gametes. This generative cell divides into two male gametes. So male gametes are nothing but reproductive cells. So how many cells now here this pollen grain contains? 1, 2 and 3. If the pollen grain shed at this stage, this is known as 3-cell stage of shedding of pollen grain. Okay, and now this pollen grain is going to pollinate by insect or any abiotic components. Okay, it will be landing on the stigma of flower, and then the germination will be taking place. Fine. For today, this much only of this chapter. Nothing much we are going to discuss in this video, because there is so much information, so much concepts. So first learn, understand this what I have discussed in this video. I hope the concepts are clear as much as possible. The facts I shared with you, still possibility that some concepts may left, some facts may left. If I find something like that especially important for the board, I will definitely share with you. Please see this whole video, jot down all the important things I have discussed on the board. It is written, okay, and understand, learn these things. It will definitely help you. Okay, if you like this video, please do like button, and please share this. Okay, thank you so much for today.